Hello, everyone, and I'm very pleased to be here in this illustrious company, and I think it's going to be an enjoyable evening. Before I get started with my little talk, I have a little housekeeping details. We're going to have each speaker come up, and as we switch out computers, we'll have an opportunity for one question while we're doing that. And if everyone who wants to ask a question would go to the microphones or shout out their question very loudly so we can all hear it and enjoy it. Then we'll have a much longer question and answer period at the end. And so without further ado, I'm going to ask for my slides to go up. So I'm going to be talking about balancing broken brain circuits and how we do that with deep brain stimulation. First, just to disclose, I do research, teaching, consulting for the two companies that make these devices, and that's going to conflict with what I'm going to talk to you about today. So obviously, an electrician has to understand the wiring, the connections inside your house, outside your house, in order to try and fix something. And so to do that, they create wiring diagrams so they can see what's connected to what and where a problem could be. Well, we can do that same type of thing with the brain. So what you see here is a wiring diagram of the headlight of a motorcycle, and then on the left, the wiring diagram of your visual system. So we can plot these things out and start to tease out where a problem might be and see if we can help with problems like that. We have new tools that are allowing us to see the brain at work. We can see through FMRI and PET scans the brain as it consumes the nutrients it needs to do that work. So we can see oxygen and glucose being consumed, and that pinpoints an area in the brain that is working very hard for that particular instance. In addition, we have other techniques through MRI called DTI, and this shows us the cabling systems that link these areas. In addition, we have techniques that have very fine time resolution, so that we can see things in microseconds, so we can actually see a thought race across the brain. So this is an example of a PET or fMRI. It's looking at how the brain is utilizing glucose or oxygen, and it's this particular brain, which belongs to a particular person, is in the scanner, and he's being asked to look at a word, think about what it means, and then generate a word in response. And so what you can see here is the occipital cortex, that's the reception of the visual information lighting up. And over here we can see the area of the brain that is trying to wrestle with those words. But we need to know, how are those two areas connected? And so with this DTI maps, we can see those cables that go between those two areas. And we know that we have to preserve those if we want to preserve function. In this diagram, we're looking at the thought actually traveling across the brain. So here you see a tiny dot as the person first starts to look at that word. And then this area over here, if I can get my mouse to work, starts to light up as the person comprehends that word. And now, as time goes on, they start to generate the word in this area. So we can see the thought go across in microseconds. So this type of imaging has given us new insight into human behavior, cognition, emotion, so that we can now visualize the circuitry of laughter and the anatomy of sadness. But sometimes these circuits break. And this can occur through disease or injury. And so I'm going to talk about one of those times when these circuits break. I'm going to use the example of movement disorders to show you how this knowledge of circuitry can allow us to understand how to go about fixing the patient. This system requires the involvement of the cortex, our thinking part of the brain, but it's really about the deeper structures of the brain here, called the basal ganglia. And these are areas of the brain that process all our movements so we don't have to think about it. And so this creates a characteristic problem where the patient has to think about their movements if they want them to work properly. And you can only do that for a certain amount of time. This first video shows you a young gentleman. Looks like he should be able to do this finger tapping very rapidly. And if you look at me as I do my finger tapping and I'm talking and I'm looking around the audience, my finger taps never waver in the amplitude or the speed or the rhythm. It's just automatic. It's all happening in those deep basal ganglia structures. But when those deep basal ganglia structures break, then that rhythm breaks down and the movements get small and slow. And that's Parkinson's disease. We also see in these same patients that they develop a resting tremor. And here is a motor program going on that shouldn't be going on. And so they have to wrestle with their arm moving many times a second or a minute, and that can get to be very tiring. In addition, there's a funny walk that develops. Walking, if you think about it, that's a really intense automatic motor program. We joke about not being able to chew gum and walk at the same time. Well, for these folks, this is very, very real. They have a characteristic stooped posture, and they get stuck at things like doorways. Even though there's no actual blockade, their automatic motor programs think there is. And so they have to kick their motor program out of that non-thinking part into their cortical part where they're thinking. And so if they step over a cane, then that gets them out of that stuck position. We have medicines that, of course, can help these patients, and it does initially, but later on, it can create its own set of problems. These extra movements, what I call the Michael J. Fox wiggles, which is too much movement from the medicines that we're giving them. All the patients will tell you that those extra movements are infinitely preferable to being frozen, and so they continue to take those medicines. So what's this all about? Well, again, we know this is coming from circuits in the brain. And if we look at these structures, those are those deep basal ganglia structures. And what happens is this area right here, the substantia nigra, has a loss of dopamine. And that goes away. And as it does that, there are changes in these circuits. These nuclei become overactive. They put the brakes on the thalamus and the cortex, and the movements slow down. So that's the end result of the loss of this. And you see this patient, again, having those very characteristic slow movements. Okay, we know where the circuit is. We can see it's broken. But what do we do about it? We need a tool, like this screwdriver, to try and interact with that circuit. And as you might imagine, you have to have a very effective tool and one that can do a lot of things if we're going to fix all these different broken circuits. Well, this deep brain stimulator is actually an adjustable, reversible off switch. We may someday know how to make it an on switch, but right now we're really good at turning off structures in the brain, restoring balance when something is too heavy-footed, like those brakes in the basal ganglia. If we can shut those down, now we can restore balance and we can improve function. And that's what the deep brain stimulator is. It does have to go deep in the brain, hence the name, and it has a battery pack on the chest that provides that electricity and is the smart portion of it. So what does that do? Well, if we have this Parkinson's patient, if they lose their dopamine, what we can do is put in a deep brain stimulator in these two, either one of these two nuclei, and what we'll see is a restoration of that normal function. Because now the brakes are not on the cortex, and you see this same gentleman now able to tap his fingers rapidly and with the same rhythm and amplitude. So this deep brain stimulator can be very helpful. But, as you might imagine, that exact location is critical. It's like real estate, location, location, location. And so what we need is some way to get there. Now think about it. You're looking at a closed head. You got to get deep inside the brain. It's not like you can see it. So you need something like GPS for the brain so that we can go through pathways that won't harm the patient and get to the right spot. But when we get there, we're within 2 to 3 millimeters of where we want to be. And that's not good enough in deep brain stimulation. We have to be able to fine-tune that location further. And so we have 2 techniques. We have test stimulation, which is essentially tried it on for size, and microelectrode recording, where we listen to the brain cells. And believe it or not, we can tell where we are by listening to those brain cells. So this GPS for the brain used to look like this. Looks sort of like an astrolabe or something from the ancient mariner times. Well, this is the new technique, which is frameless stereotaxy. So we use a lot of computers, infrared lights, et cetera, that sort of thing. And in doing so, we now free up the patient's face and head. We don't have them pinned to the table. And we can do this test stimulation. And it's much easier to see how the patient's interacting. And so we have this gentleman who had a tremor. We turn on the stimulator, and now we can see that tremor disappear right there on the operating table. And so we know we have a good spot to help that patient. But in addition, we have another technique, which is that we're listening to these brain cells. And as I told you, we're going to try and turn something off. So it makes sense that the thing that we're going to turn off is something very overactive. And indeed, that's what it sounds like. As we're going through the brain, you can hear some cells, and it's nice and quiet, and then all of a sudden you hear a dull roar in the background. And as you approach this nucleus, it gets louder and louder and louder. And this is a little bit about what it sounds like. So it sounds like static, but it's a whole bunch of cells, all firing in all different sorts of rhythms, and you get a lot of noise out of it. So that tells us, okay, we're in roughly the right spot. But now we have to figure out if we're in the spot that's actually tied to the motor system that we want to work on. And so we then look at these rhythms here, and as we come to the part that's tied in with the motor cell, say of the arm, if we move that arm, we'll hear those motor cells change in rhythm with the movement of the arm. And I'll show you that. In addition, if we come to this lower structure, which is the optic tract, that's taking the information from the eye back to the brain to perceive light, we can actually hear that traffic go through that nerve. And so if you listen carefully, you'll see what I'm talking about. Here's a gentleman who's having his arm moved. You can hear the rhythm change with that. And that little brush is the light going through that optic nerve. So what does that do? This is all very fine and good, but how does it help the patient? Well, here's a gentleman who, before surgery, has Parkinson's disease. He's barely able to get out of the chair. He's going to try and walk down the hallway, turn around, and come back. And this is the same gentleman with no medication, just stimulation, who's walking down the hallway afterwards. And what you'll see is that post-op, he goes up and down that hallway 5 times in the same time that it took him to do it once preoperatively. So we can have a really powerful effect with this deep brain stimulation. But we can also treat other movement disorders. So here's a gentleman with tremor. This person happens to have Parkinsonian tremor, which is a resting tremor, but we have the same effect on essential tremor, which is a tremor as you come to try to use something like drinking a cup of coffee, your tremor will get worse. And so now you'll see him after his surgery. He's got the device turned on, and what you'll see is now he has steady hands, and he's able to take that cup of coffee and take a sip from it, and he'll even give us a cheers. Nice and steady hand. But my favorite disease to treat is this dystonia. This is a horrible disease. It takes a normal kid and turns him into a pretzel with these very abnormal motions. This is a young man who started out very normal, and over the course of his late childhood developed these movements, and now he wasn't able to walk. He was either in a wheelchair or he'd crawl around the house. Hadn't walked for several years. And so we did the deep brain stimulation, again in these deep parts of the brain, and now just 1 month later, he's able to walk down the hall. You can see he still has a little bit of a tilt that takes some more time to go away. And so we send him off. He becomes a masseuse in a cruise ship. I thought that was pretty good use of his time. And so he doesn't come back for his 1-year follow-up. He comes back for his 2-year follow-up. And now we see that not only has he gotten his normal motions back, but he's become a very normal young man. He goes on to get married and has his life restored to him. So these things are very powerful. So deep brain stimulation is also being used for the treatment of depression. This is experimental. This is not yet developed. It's being looked at for dementia, a very important area. Obesity, another very important area. Epilepsy, but we're out of time. So just one more thought for you. Remember the matrix. It is all in your head. Thank you very much. Appreciate it.